Life did not ask for permission to exist on Earth. It showed up in boiling volcanic pools. It colonized the pitch-black ocean floor. It tunneled 3 kilometers into solid rock and kept going. And the universe, it turns out, has been running the same experiment, the same chemistry, the same ingredients, the same conditions, across 2 trillion galaxies for nearly 14 billion years. This is not a story about belief. This is not a documentary about blurry photographs and government cover-ups. This is a case. A logical, evidence-based, scientifically documented case. And by the time we reach number 1, you are not going to wonder whether alien life exists. You are going to wonder why we ever thought we were the exception. Let's count down. Number 5. The phosphine signal on Venus. Venus is not supposed to be interesting. It is 465 degrees Celsius on the surface. The atmospheric pressure would crush a submarine. Clouds of sulfuric acid cycle through skies so thick that sunlight barely reaches the ground. For decades, Venus was the textbook example of a dead world, a cautionary tale about runaway greenhouse effects, not a candidate for life. Then, in September 2020, a team led by astronomer Jane Greaves published findings in Nature Astronomy that stopped the scientific community cold. They had detected phosphine in Venus's atmosphere. Not a trace. A measurable, confirmed concentration sitting in the cloud layer approximately 50 kilometers above the surface, where temperatures drop to a relatively mild 30 degrees Celsius and atmospheric pressure resembles Earth's surface. Here is why that matters. Phosphine is a molecule made of 1 phosphorus atom and 3 hydrogen atoms. On Earth, it is produced in 2 ways: industrial manufacturing, and anaerobic bacteria, microorganisms that live without oxygen. There is no industrial manufacturing on Venus. And every geological and chemical process scientists attempted to model fell orders of magnitude short of explaining the concentration detected. The researchers ran every abiotic scenario they could construct. Lightning. Volcanic activity. Photochemical reactions. None of them came close. The only known process that produces phosphine at that concentration on a rocky planet is biology. Ask yourself what that means. Not on a distant star system. Not in another galaxy. On the planet that is, right now, the second dot from the sun in your night sky. Number 4. Enceladus is venting the ingredients of life into space. Saturn's moon Enceladus is 500 kilometers wide. That is smaller than the state of Texas. For most of its discovery history, it was considered geologically dead, too small, too far from the sun, too frozen to be anything but a snowball drifting through the outer solar system. The Cassini spacecraft changed everything. In 2005, Cassini detected enormous plumes of water vapor erupting from cracks near Enceladus's south pole, geysers blasting hundreds of kilometers into space, feeding directly into Saturn's E-ring. Scientists confirmed a liquid water ocean beneath the ice, heated by tidal forces generated by Saturn's gravitational pull. Then the real discovery arrived. In 2017, analysis of Cassini's plume data published in Science confirmed the presence of molecular hydrogen, H2, in concentrations that could only be explained by active hydrothermal reactions on the ocean floor. Hot water. Mineral-rich rock. Chemical gradients. The exact conditions found at hydrothermal vents on Earth, the same vents where life thrives without a single photon of sunlight. In 2023, researchers announced the detection of phosphates, key components of DNA, RNA, and cell membranes, in the plume water. Let that register. A moon the size of Texas, hiding beneath its frozen crust a warm saltwater ocean with active geology, organic chemistry, hydrogen fuel sources, and the molecular building blocks of genetic material, actively spraying that material into space for any passing spacecraft to sample. We did not have to drill. We did not have to land. Enceladus handed us the evidence. Number 3. The James Webb Telescope found carbon in exoplanet atmospheres. For the first time in human history, we can read the chemistry of other worlds. The James Webb Space Telescope, the most powerful observatory ever built, detects light passing through exoplanet atmospheres as those planets transit their stars. Different molecules absorb different wavelengths of light. Webb reads those signatures like a fingerprint. In 2023, Webb analyzed the atmosphere of K2-18b, a planet 8.6 times Earth's mass, orbiting in the habitable zone of its star, 120 light-years away. It found methane, carbon dioxide, and a tentative but statistically significant signal for dimethyl sulfide, a molecule that, on Earth, is produced exclusively by marine phytoplankton. No known geological process generates dimethyl sulfide. No known chemical reaction without biology produces it in detectable quantities. The researchers were careful. They published. They noted the signal required confirmation. But the data was real, the methodology was sound, and the molecule was there. Run the logic. A planet in the habitable zone. A liquid water ocean likely beneath its atmosphere. Carbon dioxide and methane in concentrations consistent with biological activity. A biosignature molecule that life on Earth exhales into the ocean air every single day. This is not a blip. This is a telescope pointed at a specific planet, detecting a specific chemical, that specific chemistry does not produce without life, 120 light-years away. Number 2. The universe is manufacturing life's ingredients in empty space. Here is what they found drifting in the molecular cloud Sagittarius B2, a massive gas cloud near the center of our galaxy, in the empty void between stars, nowhere near any planet, nowhere near any source of liquid water, glycolaldehyde. The simplest sugar molecule. A direct precursor to ribose, the sugar backbone of RNA, the molecule that carries genetic instructions in every living thing on Earth. They found it using radio telescopes in 2000. They found it again in multiple other molecular clouds across the galaxy. Then, across decades of observation, the list grew. Formamide. Acetaldehyde. Amino acid precursors. Complex carbon chains that slot directly into the machinery of biology. The universe is not waiting for a planet with the right conditions to start building the chemistry of life. It is building that chemistry in open space. In the cold, irradiated void between the stars. Spontaneously. Constantly. Across every corner of the galaxy we have pointed a telescope at. Every rocky planet that forms, every moon, every asteroid, is assembled from this preloaded chemistry. Earth did not generate the raw materials of life from scratch. It inherited them. From a universe that has been running organic chemistry experiments for longer than our planet has existed. There is nowhere in this galaxy where the ingredients of life are not already present. The question was never whether the materials existed. The question was whether assembly ever occurred, and that question has one answer, because you are reading this sentence. Number 1. We have already found it. We just haven't accepted it yet. In 1996, NASA scientists announced findings that shook the world for about 48 hours before being quietly buried under skepticism. ALH 84001. A meteorite recovered from Antarctica. Confirmed Martian origin, its chemical signature matched the Martian atmosphere precisely. Inside it, under electron microscopes, researchers found structures. Elongated, segmented, roughly 100 nanometers long. Structures that looked, unmistakably, like fossilized nanobacteria. They also found magnetite crystals arranged in chains, a formation on Earth produced exclusively by magnetotactic bacteria. They found polycyclic aromatic hydrocarbons, organic compounds associated with biological decay. They found carbonate globules consistent with biological processes occurring in liquid water. NASA held a press conference. President Clinton made a statement. Then the scientific community spent the next decade dismantling it, not disproving it, dismantling it, finding alternative explanations for each piece individually, even as no single alternative explained all pieces simultaneously. The debate is not settled. It never closed. And here is the question that should be keeping you awake tonight. If every individual piece of that meteorite's evidence has a potential non-biological explanation, why is it that when you assemble all of them together, the structures, the magnetite chains, the organics, the carbonates, the simplest, most parsimonious explanation is still biology? Because in science, when every piece of evidence points in one direction, that direction tends to be correct. We found a rock from Mars. Inside it, something made structures, chemistry, and arrangements that life makes. We chose not to accept it, not because the evidence was absent, but because the conclusion was too large. So here is the real question. Not whether life exists elsewhere. We have covered that. The real question is this: if life did exist on Mars billions of years ago, before Mars lost its atmosphere, before its oceans evaporated, and Mars and Earth have been exchanging meteorites for 4 billion years, where, exactly, do you think Earth's life came from?